this talk, I will focus on probabilistic factor models, which I think are a very useful tool for the interpretable integration of multimodal omics data. And already the previous talk has touched on these multi-omics data. So with multi-omics assays, we try to jointly study different classes of molecular layers, for example, genomics, proteomics, transcriptomics, on the same set of sample. And this will help us to get a more comprehensive quantitative characterization of complex biological processes and traits. And the resulting data leads to these multimodal data set collections where we have different data sets from each of these different omic technologies, and possibly also for different groups of samples, for example, from different experimental conditions. And one area where this is of increasing interest, we've also seen in the previous talk, is personalized medicine, where we try to better understand the heterogeneity across patients in terms of disease progression and treatment outcome. And for this, these multi-omic profiling studies promise us to gain a better understanding of this heterogeneity on a molecular basis that looks at all of the molecular layers, and thereby eventually enable more personalized treatment decisions. However, the challenge here is to integrate these diverse data modalities in an interpretable manner, and thereby also be able to go back with the results of our analysis to domain experts or clinicians and take actions. And for this purpose, we've recently developed multi-omics factor analysis, which is a probabilistic factor model that performs a structured matrix factorization. So the idea is that we decompose our input data, which consists of these different views, which are the features measured from different molecular layers, and possibly also different sample groups, for example, from different experimental conditions, into a set of factors that are joined across all of the different views and represent sort of the major sources of variation in the data. And we link those latent factors back to the original data via weight matrices. And importantly, we pose sparsity priors both on the weight matrices and on the latent factors to improve the interpretability of the model. And I will come to this also in more detail on the next slide. As this is a Bayesian model, we want to infer the posterior at the end. And for this, we use stochastic Bayesian base, which makes the model very scalable also to hundreds of thousands of samples. And so this is an overview of the Bayesian graphical model that analyzes framework. So we model our observed data, which is a gray node Y here, as coming from some distribution, often simply a normal distribution, where the mean parameter is given by a product of the factors, which are per group, and the weights, which are per view. And as I said, we use those sparsity priors, where we combine an automatic relevance determination prior, which tells us for each factor how important it is for a certain view, so omic layer, and for a certain group of samples. It's a spike and slab prior, which is the second part here, which modulates the feature-wise or sample-wise sparsity of the factor so that we can more easily pinpoint individual molecular features that are responsible or driving the variation that is behind one of the factors. So these sparsity constraints really help us to answer questions like which modalities are important for a factor, which factors are important in which sample groups, and also which features, molecular features, are important for a factor. And we've applied this method to a data set from chronic lymphocytic leukemia patients, where we try to better understand the sources of heterogeneity in blood cancer. So we had a set of 200 leukemia samples, which were incompletely characterized by 4 molecular assays, namely genomic sequencing, RNA sequencing, methylation arrays, and ex vivo drug response assays. And for this, we recovered 10 factors using the MOFA framework. And you can see that there's a very clear Bayes decomposition, where we can distinguish factors that only explain variation on some of the molecular layers from factors that are more universal, like the first one, which is active on all of the different molecular layers. And then we can look into the weights of the factors in each of the different molecular layers. And thanks to the spike and slab prior, this often highlights a small number of features that are important here. And just as one example, I here illustrate the first factor, which if we visualize the scores of the samples along this factor, we see that this nicely recapitulates known clinical subgroups, which are illustrated by the colors here of this disease. And we can link these to the different molecular layers. So for example, the genomic marker IGH3, which is used to define the clinical subgroups, but also other molecular layers, for example, identifying differentially expressed genes, or also drugs that show a differential response depending on where the sample lies on this factor. And we can not only calculate these clinical subgroups, but we can also refine it, so get a continuous score instead of just a binary classification. And we can identify outlier samples, like, for example, the red sample down here, where the clinical annotation is not in line with the molecular multi-omic profile. And we've also showed that if we take all the 10 factors together to predict clinical outcome of the patients, we can do much better than if we would only look in one of the different omic layers individually. However, one drawback of this approach that we've noted is that such kind of factor models do not account for spatial or temporal relationships between the samples that, however, are more and more common to have in some of the studies. For example, in precision medicine, there are now a number of longitudinal studies where a patient cohort is followed over a time period, and these different multi-omic measurements are taken at each of these time points during, for example, 2 years. Also in developmental biology, we of course have a clear time component. If you, for example, study gene expression over development, and also in single cell biology, we now more and more start to use spatially resolved approaches to study a cell in its natural context and not out of it. And in such kind of data sets, we of course again want to integrate these different data modalities, but we also want to account for the temporal or spatial dependencies that are naturally present in such between the samples in such data. And so we would like to understand which sources of variation are linked to space and time and how they differ between individuals or species. And for this purpose, we've extended the sparse factor framework I shown you previously with a Gaussian process prior in the latent space to better identify spatial or temporal patterns from such multimodal data. So the input data is similar as previously in the MOFA framework. We have these different omic layers measured on a set of individuals, which are groups of samples, but in each of these groups, we now have a temporally resolved or spatially resolved trajectory along which we take samples. And then we decompose this data again into latent factors, which are, however, now are seen as a continuous factor that's modulated by a Gaussian process, and therefore help us to better distinguish temporal variation or spatial variation, which are the first two components here, from non-temporal variation, and also distinguish shared temporal variation that is common across all individuals from group-dependent temporal variation. So again, we use priors on the weights to promote the few and feature-wise sparsity and again interpretability as in the first framework, but we now have a Gaussian process prior on the latent factors that model both the temporal covariance structure between the samples using a squared exponential kernel and also a group-group covariance structure that tell us how the different individuals are related. And using these learned covariance structures, we can then do novel downstream analysis, like, for example, interpolating or extrapolating to missing time points or spatial locations, and also cluster the individuals or the groups of samples on this individual level, or identify outliers based on the temporal or spatial patterns. And we've applied this method to an evolutionary atlas of gene expression, where gene expression was profiled in multiple species along development in multiple organs. And here we had an additional problem that the developmental time points between the species did not match, so we didn't know the correspondences between them. And therefore, we implemented the method to jointly learn the latent space, as well as do an alignment based on dynamic time warping to match the time points between the different species. And I just want to show you a quick glimpse at the results we got here. So we could infer 5 latent factors, and based on the Gaussian process hyperparameters for each of them, we could calculate the smoothness and a sharedness score, which sort of indicate us how smooth the variation captured by the factor is along time and how shared it is across the different species. And in this example, all of our factors had a very high smoothness score, which is to be expected because in such a developmental data set, we hope that most variation is driven by developmental programs. And the sharedness score, however, varies between the different factors. So we have 3 which have a high sharedness score, so have a very strong group-group correlation, which are capturing conserved developmental programs, while we also have 2 factors that have a lower sharedness score, where we have distinct patterns for humans and opossum, which makes sense because these species are much more evolutionary distant from the other 3 species. And now we can, of course, start looking into the weights of each of these factors and doing looking into the molecular mechanisms, which I don't have time to go into here. So I just will conclude for this talk and just summarize. So I've shown you that with MOFA, we've developed a scalable Bayesian factor analysis framework that is able to integrate structured multi-view and or multi-group data in an interpretable manner using these sparsity priors. And with Mephisto, we've extended this framework to model also temporally or spatially solved multimodal data using Gaussian process priors in the latent space. And thereby we can distinguish shared or specific patterns and also align time points if we have imperfect correspondences between the different groups. And modeling these covariance structures in the latent space is especially helpful in data sets where we have many missing values, because here it allows for an extra interpolation of the temporal and spatial trajectories in the latent space. And the software is available from GitHub or the R part also on Bioconductor. And if you're interested in more, get in touch or have a look at our webpage for MOFA and Mephisto. And with this, I'm at the end of my talk and just would like to acknowledge all the people who contributed to it, both from our lab at Olli, but also at EMBL and external collaborators, Thorsten, Sascha, and Margarita.
Thank you, Britta. We have time for maybe one or two quick questions, if there are any. Fabian asks one question.
Sorry, Olli, this was still discussion.
The old question, I see. Okay, great. All right, sorry. Ongoing discussion about the previous talk. I got confused here. Stefan Reichel asked a question, and he was wondering how Mephisto does deal with very few, for example, 3 non-equivalent timescales or time points per patient. So yeah, I guess lots of missingness and difficulties in aligning them essentially.
Yeah, so I mean, the missingness is one thing that Mephisto takes better care than the MOFA framework, because MOFA relies only on feature correlations to construct the latent space, while Mephisto also takes the time information into account, and thereby it can better handle with missing values and also interpolate them. The alignment requires, I'm not sure where sort of the limit is, how many time points you need that the alignment still works. In our EvoDevo data, we had quite a lot of missing stages for the different species. So we had between 11 or 21 time points for each of the species, and there the alignment worked really well. With 3 time points, it can be difficult, I guess, but I would have to investigate that.